Welcome back to Chapter 6, Lecture 4. And in this section, we're going to talk about enzymes. I know that y'all have already done some work with enzymes in lab, so some of this may be a review, and that's fine. Repetition is always good. So what are enzymes and what do they do? We know from things that we've talked about in lecture already that most enzymes are proteins, but not all. However, what we're going to talk about in this lecture and the next one is primarily dealing with enzymes that are proteins. That's the most common occurrence. So unless I say specifically, like in the ribosome example, the enzyme is the RNA and not the protein, if I'm just talking about enzymes in general, we can assume we're talking about proteins, which means that if we're talking about proteins, you've got to go back and think about all of the characteristics of proteins that we learned earlier in the semester. So they're polymers of amino acids. They've got primary, secondary, tertiary, and sometimes quaternary structure. What are the stabilizing forces of those different levels of protein structure? All of that's going to be important when we talk about how enzymes do their jobs, okay? But the most important thing about enzymes is that they lower the energy of activation for chemical reactions that take place within cells. Okay, it's important to know a couple of things. Enzymes do not provide energy, and they do not change the delta G of the reaction. So if you look at this figure, the black line is showing us the course of the reaction without the enzyme. So we see that this is an exergonic reaction like we talked about in previous lectures. We see that the reactants start higher on the free energy scale than the products, and that the reaction goes uphill and then comes down to products. So we see this energy of activation without the enzyme. When you add an enzyme to that reaction, the red line shows us what happens. The activation energy is decreased, which allows the reaction to go faster. So that's another really important quality about enzymes, is that they speed up reactions. And they do that by lowering the energy of activation, okay? Another way to think about them is that enzymes are biological catalysts, okay, which is basically saying the same thing. They speed up chemical reactions because they are catalysts, okay? But notice the delta G for both reactions with the enzyme and without the enzyme is the same, because the enzymes are not changing the free energy of where the reactants start, and they're not changing the free energy of where the products finish. They are only changing the energy of activation. All right, well, before you freak out when you look at this image, in a cell, enzymes control metabolism. So remember a couple of lectures ago, we talked about the fact that metabolism is the total of all the chemical reactions that take place in an organism. Well, this is a simplified diagram of human metabolism. And so I know it's really, really tiny, but if we zoom in here a little bit, every dot on this chart represents some kind of a chemical intermediate, some kind of a molecule. Every line connecting two dots represents a chemical reaction. Every chemical reaction, maybe I shouldn't say every, almost every chemical reaction in a cell requires an enzyme. Okay, so look at all of these dots that are connected by lines. Enzymes are critically important for controlling human metabolism. They make those reactions go faster. Without enzymes, those chemical reactions would not happen at a rate that would be sufficient enough to support life. Okay, so really, really important. We've talked a lot about how proteins are the workers of the cell. Enzymes are really the cog in that worker wheel. Okay, you can't have living cells without enzymes. Okay, let me try to get this back where I can go to the next slide. There we go. Okay, so how do enzymes work? Enzymes have active sites where their substrates bind, and hopefully you've already talked about this in lab, okay? And the current model for understanding enzyme substrate binding is called the induced fit model. All right, and if we're going to induce something, we're going to cause something to happen. Okay, if someone is pregnant and they go in to be induced, we're going to cause labor to happen. So if we're going to induce the fit, that means we're going to do something that's going to cause the fit to happen. Well, if we look over here at part A of this figure, we see an enzyme. It's in its tertiary structure, and it has an active site right here. Okay, that active site is going to be specific for the substrate to bind. Okay, the substrate is going to be a reactant in a chemical reaction. Well, notice when the substrate bound the enzyme, this enzyme shape changed a little bit. Okay, look at like this part of the enzyme right up here, and you compare it over here, it has shifted down. Okay, so the way I like to think about this is that when the substrate binds the enzyme, it basically induces the enzyme to hug it, okay, or snug its fit down around that enzyme. That's really important because that induced fit is what facilitates lowering the activation energy. All right, so if we're going to have a chemical reaction and we're going to convert reactants into products, then we're going to have to break chemical bonds. Okay, we're going to have to break covalent bonds. And covalent bonds are the strongest of the chemical bonds. Well, think about it this way. When you were a kid and you were playing outside in the fall and you picked up a stick that had fallen on the ground and you wanted to break that stick in half, you didn't just put your hands on each end and pull it apart like the Hulk, right? How did you break it? You bent it over your knee, okay? You bent it. That made it easier to break. Well, enzymes do the same thing with the chemical bonds in substrates, okay? When the enzyme induces its fit around the substrate, it is going to either bend chemical bonds or twist them or otherwise stress them in some way. And when that happens, when those bonds are bent or twisted or stressed, it takes less energy to break them. And that is how the enzymes are going to lower the activation energy of a chemical reaction, is by stressing the chemical bonds. But they cannot stress the chemical bonds in the substrate if they cannot induce their fit around the substrate. Okay, that is really important. The other thing to know about enzyme substrate binding is that it is specific. Okay? Enzymes typically only bind to a particular substrate. Okay? So a substrate that or an enzyme that recognizes glucose is not going to recognize fructose because they have different shapes. So remember our other Bio 1 mantra, shape determines function. Okay? An enzyme's active site is specific to the shape of its substrate. That's really critical. All right, so when we think about induced fit and we think about the flexibility that enzymes have to be able to do that, it's just a good time to come back and review quickly what those stabilizing forces are in protein structure. Okay, so remember, secondary structure of proteins, we're talking primarily about beta sheets and alpha helices. And these are stabilized by hydrogen bonds between the amino acid backbone atoms. Okay, so for secondary structure, we are not talking about the R groups. For tertiary and quaternary structure, though, remember, we are talking about the R group. So the overall tertiary and quaternary structure is determined by protein folding which is directed by the R groups. And the stabilizing forces of tertiary structure are going to be hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bonds. And remember, disulfide bonds, these are the only covalent interactions for tertiary and quaternary structure. Okay? And remember, all of these are taking place between R groups. Well, why is this important? It's important because hydrogen bonds, ionic bonds, hydrophobic bonds, they allow for protein flexibility. Because these bonds are relatively weak, because they're not like setting the protein, we're not casting it in bronze where it's completely hard and inflexible. These interactions that stabilize tertiary structure allow the protein to flex and to move and to, just change its shape slightly. When a protein changes its shape, that's important, okay? Sometimes proteins change their shape and they don't work anymore. But for an enzyme, when it induces its fit around that substrate by changing its shape just a little bit, that is how enzymes lower the energy of activation. That's how they can make reactions go hundreds or even thousands or even millions of times faster than normal, okay? Because they can change their shape and induce their fit around those substrates that stress the chemical bonds of the reactants and make them easier to break. All right, well, another really interesting thing about enzymes as catalysts is that they're not used up by the reaction, okay? So the catalytic cycle of enzymes, notice the word cycle, okay? An enzyme, if we look at this figure, we have an enzyme that is binding its substrates, okay? When that happens, that's called the enzyme substrate complex. That's when induced fit is going to happen. Okay, so the enzyme stresses the bonds in the substrate. It makes them easier to break, which lowers the energy of activation, which speeds up the reaction. Okay, the products are going to be created, and then those products are going to be released, and then the enzyme returns to its original conformation, and it's ready then to pick up more substrates and do the same thing over again. So notice in this drawing how the enzyme without substrates has a very different shape than the enzymes after substrates have bound. Okay, you can see that induced fit, how the enzyme kind of hugs those substrates to stress the chemical bonds. So how important is this? You have worked with catalase in lab. Okay, one catalase enzyme can do this cycle 1,000,000 times or more in 1 second. Okay, so 1001, 1 second, and a catalase enzyme can do this 1,000,000 times. That's crazy fast. That's why enzymes are so important in cells, because there's no way that those reactions could go that fast without enzymes. That's hard for us to wrap our minds around, okay? And catalase is a unique enzyme. It's one of the most efficient enzymes. Most enzymes don't have rates that are 1,000,000 times a second. Okay, catalase is kind of unique in that way, but it just gives you an idea of how important those enzymes are. Now, because enzymes are proteins, they can be affected by their local environment. Also, different organisms that live in different environments, their enzymes are specially suited to those environments. So temperature and pH are two environmental conditions that can really affect enzymes. So if we look at this top graph, the black line represents the optimal temperature for a human enzyme. Okay, and notice that optimal temperature is 37 degrees Celsius. 37 degrees Celsius is body temperature. Okay, so our enzymes are going to work best around body temperature. Well, what happens if we increase the temperature where those enzymes are by about 6 degrees and we keep it there for a while, excuse me, those enzymes are going to denature. They're not going to work anymore. And it's because as you increase the temperature, you are destabilizing the interactions that hold the protein together. So those hydrogen bonds, those ionic bonds, they cannot hold the protein together at higher temperatures. What happens at higher temperatures? Things are moving faster, there's more energy, can't hold those bonds together, and so the tertiary structure is going to fall apart. It's going to unfold. And when a protein denatures, okay, it cannot do its job. Well, if you look on the other side of the optimum for humans, okay, on the cold side, cold temperatures don't cause enzymes to denature, but they do cause everything to slow down. And what happens when you freeze something? Is it flexible anymore? Not usually, okay? So when you cool down enzymes, they become inflexible. If they are inflexible, what can't they do anymore? They can't induce their fit around the substrates, so they can't do their jobs. And so the rate of the reaction that they're catalyzing goes way down, okay? If you warm them back up, they'll regain their activity. But if you heat an enzyme past its optimum and it denatures, cooling that enzyme back down is not going to restore the activity. Because when an enzyme denatures, it can't just fold back spontaneously. Protein folding is hard, and that's just not going to happen. Okay? Now, if you look at this red line, here we have a thermophilic bacteria, a heat-loving bacteria that has an optimum temperature of 75 degrees Celsius. Put that in your cell phone temperature converter. 75 degrees Celsius is really hot, right? Boiling for Celsius is 100, boiling for Fahrenheit is 212, so you kind of get the idea. It's really hot. Human enzymes are long since denatured at that optimal temperature. But for these heat-loving bacteria, at the optimal temperature for human enzymes, they're too cold. They can't function either. Okay, so these organisms and their enzymes are evolved to work in the optimum environment for where they live. Okay, so down here on the bottom, we've got the effects of pH on enzyme activity. Both of these are human enzymes. Pepsin works in the stomach. Well, what do you know about the stomach environment? It's really acidic. Pepsin works best at a pH of 2. Well, the stomach is really about the only place in the human body where you're going to have pH of 2. That's crazy acidic. Trypsin, on the other hand, works in the intestines, and it has an optimal pH of 8. On either side of those optimum pHs, the enzymes will begin to denature. And it's because when you change the pH, you are changing the ionization states. Let me just write that. So remember, our acidic amino acids have a negative charge, and our basic amino acids have a positive charge because they've either gained or lost hydrogen ions. Well, when you change the pH, different chemical groups are going to lose or gain hydrogen ions, and it's going to disrupt those hydrogen bonds and ionic bonds that hold the tertiary structure together. So changing the ionization states is another way to destabilize those interactions that hold tertiary structure together. Okay, so temperature and pH are just two examples of environmental conditions that can affect enzymes.